Research Institute. Since 2004, I guess, when we launched the Center for Math Medicine in the field, the idea was to provide a sort of nexus for people in the medical, biological, biomedical sciences and the mathematical sciences. And in many ways, it has sort of fulfilled that role. But as we found with sort of on-site seminars, I guess people have a case of seminar burnout, and audiences tended to fluctuate. But I'm sort of amazed to see, Carmen, that we've actually got 34 participants so far, and so I think this may be something we'll do on a more regular basis for the Center for Math Medicine seminars. And anyway, so at this stage, without taking up any more time, I'll hand over to Francis to formally introduce Carmen.
Okay, so thank you very much, Siv. And as Siv mentioned, this is our inaugural virtual online CMM KCN talk. And so I'm very pleased that Carmen accepted our invitation to be that person. But I mean, I think the advantage, excuse me, as mentioned by Siv, is that we can get an audience sort of in more locations. And when you have interdisciplinary topics and stuff like that, it's really important to have many different people from many different fields. So I think this is certainly something we might want to try to continue in a virtual basis. So thank you for the support from Fields and Center for Mathematical Medicine for these KCN events, Crumble Computational Neuroscience events, in the form of these CMM Fields seminars. And just a special thank you to Sarah at Fields, who sort of got all this sort of Zoom and different things set up. So appreciate your help there. And so basically, for those of you who don't know Carmen Canavier, she's one of the sort of very prominent computational neuroscience people, and so it's been a pleasure to know her over the years. And I have to start by mentioning one of the common things we have is curly hair. And I don't know if Carmen remembers, and I don't know if it was Carmen or Avramo or Victoria, one of us curly-haired people initiated this club for curly-haired women in computational neuroscience. But of course, if you didn't have curly hair, you could still be part of it and all the rest of it. So that was many years ago. And I mean, I think part of that is that, to encourage sort of having more women, which, we need also, but it's just sort of a community, which is very important. And so that was a lot of fun. And even though my hair is long now and I pull it back, it's still pretty curly. So Carmen comes from an engineering background, chemical engineering, in Vanderbilt University. And if you guys read her bio, she has a master's and PhD in computer engineering, and she did a lot of work. I remember seeing some of her early papers in sort of finding sort of the chaotic regimes and doing tons and tons of simulations from different initial conditions. So you can essentially see the butterfly effect, having different initial conditions. And that was in Jack Byrne's lab in Houston of learning and memory. And so she's a theoretical computational neuroscientist, and her current position is now as professor and vice chair for research in the Department of Cell Biology and Anatomy in Louisiana State University Health Sciences Center at New Orleans. So I don't want to take up too much time because she has a lot of interesting things to share, except to say that, in the field of neuroscience, we really need, innovative new methods and ways of thinking. And I see Carmen as one of those people because I remember seeing her work on pulse-coupled oscillators and sort of these mapping techniques. So you really have to pay attention to the data and have the sort of technical expertise, and Carmen is one of these people who has sort of a dual focus in the dopamine world as well as in gamma and theta rhythms in hippocampus. So without any further ado, welcome Carmen. And and before I forget, the important thing to mention is if you have a question, Carmen is more than happy to answer the questions during her talk. I am going to be watching for virtual hands as well as the chat. So what I'll do is, because I'm going to mute everyone, is if you have a question, just put it then if I see several in the middle of the talk, then I'll kind of, speak up so then Carmen knows, so then you have a chance to ask. Okay, but otherwise there'll be time at the end to ask questions too. Okay, the floor is yours, Carmen. Thank you.
Thank you for that very nice introduction, Francis. And I'm going to be talking about inhibitory interneuronal networks. Some of it is collaborative work with John White, funded by NINDS, and I also want to acknowledge my NIMH funding with Sonia Gasparini. So I want to try to talk about two topics. One is about how Interneuronal networks can break up into clusters and coalesce back into global synchrony. And the other one is to talk about the different synchronization properties that type 1 and type 2 excitability might confer on interneurons. And so this is like a motivational slide here. It's a slide from a model of the thalamocortical loop based on work by Alain Destech, but implemented in this case by Andrew Knox. And it's showing the activity of, well, here, cortical neurons, thalamic reticular neurons, and thalamocortical neurons, and in a model of 8 to 10 hertz spindle activity. And what I want to point out is that during the simulated spindles, the thalamocortical population breaks up into 2 clusters. And so some of them fire on one population cycle and the others fire on the next population cycle. And so the net result is these 8 to 10 hertz spindles. And the reason I think they break up into clusters is because they're excitatory neurons and they're not connected to each other, but they are connected mutually via feedback inhibition from the thalamic reticular nucleus, which is inhibitory. And so they have feedback inhibition. And I think that is what allows them to break up into clusters. And I'm actually working on a project with Laura Schrader at Tulane now to try and apply the methods I'm going to show you to this system. But what happens when you change certain parameters and tweak them, the thalamocortical neurons coalesce into one cluster. And so now the frequency is halved because instead of having them staggered, they're all firing together. And you get 3 to 4 hertz network activity. That they're making an analogy with spike and wave discharges. But basically, this is just an introduction to the concept of why would I care if interneurons, and again, thalamocortical neurons aren't interneurons, but they're functionally connected via inhibition. So I'm going to look at networks of inhibitory interneurons. And as Francis said, I'm going to apply phase resetting methods under pulse coupled assumptions. And so in order to do phase resetting, you need an oscillator. So we need to assume that neurons are, at least for some period of time, in the mean driven regime where they fire regularly. And if you have a neuron that's firing regularly, and we're going to call the time the spike is emitted a phase of 0, and the time the spike is emitted a phase of 1, and then it's circular. So then you'll have all phases possible in between. And what I'm going to do is perturb the neuron, in this case with a bi-exponential inhibitory conductance. And the key idea is to perturb the neuron with the input that it's going to get in the circuit that you're going to study. And so Nancy Capel calls this type of a phase resetting curve a spike time response curve. I'm just going to go with a phase resetting curve. But the important quantities are the stimulus interval, which is the time between when you emit a spike and when you get an input, and the response interval, which is the time between when you get an input and you emit a spike. And these quantities are easily derived from the phase and the phase resetting curve. So the phase resetting is if I give a perturbation, does the next spike come sooner, which would make it an advance, or does it come later, which would make it a delay? And so the phase resetting is actually this normalized change in the cycle length as a result of getting a perturbation. And we call this the phase resetting. I'm going to call it FFV. So it's normalized. Now the convention, in this convention, a positive phase resetting delays the next spike. A negative phase resetting advances it. So this is contrary to the mathematical convention, and I apologize for that, but I got it from David Perkel, and I've used it consistently, so I'm going to keep doing it now. But in any case, the stimulus interval is just the phase times the intrinsic period. And then the response interval, if there was no perturbation, it would simply be the fraction of time remaining till the next spike, which would be 1 minus the phase times the intrinsic period. But then you have to add any resetting to this response interval. So these, I'm going to talk about making maps of these intervals, so I wanted to find them up front. So that's phase resetting. And so as I said, once you now we have taken an open loop phase resetting curve, and we're going to make the strong assumption that we put a neuron into the circuit, that it's now, and we use the input from the partner neuron as the input when we generated the phase resetting curve. And we're going to assume that when we put this neuron in a circuit, that it's going to respond in the same way to the inputs in the circuit as it did to the one in the open loop. That's actually a strong assumption, but we're going to make that. And then, so we have a closed loop. We have to, and I'm only going to deal in this talk with identical oscillators with identical connections, because that's going to greatly simplify the math. All right, so if we know an initial condition and we assume, well, if we know an initial condition, we can determine for the rest of time what this neuron is going to do under this assumptions. But I'm just going to show you an assumed firing pattern here, that I'm assuming alternating firing here. But so if you know the response time in neuron 2, that automatically defines the stimulus time. So this interval is the time between it fires and when it gets an input. And this stimulus time defines the response time based on because you've just determined those stimulus response pairs using the phase resetting curve. And then the response curve determines the stimulus interval in the next neuron. So you can see that you can build a map here. And the map's on the right-hand side of the screen. So I can build two difference equations in the two phases. So you've now basically strobed this system. It's a continuous system, but we're only going to look at it when a spike is emitted and an input is received, and we're going to make a map based on that. And the map's here in this lower right-hand corner. And maps have fixed points. So maps are difference equations. It's the discrete equivalent of differential equations. And their stability can be determined using the slope of the PRC is the point that I'm going to try to make here. But so we have a difference equation, and you can find the fixed points of this difference equation. And these are the points at which the phases would stay constant on each cycle and not change anymore. And so once you find this fixed point, we're going to assume a perturbation, delta phi, in the phase about the fixed point. And then we're going to linearize the phase resetting curve. So now what's going to be important is not the value of the phase resetting, but the value of the slope. And so, like I said, I'm not belaboring the math, but when you cancel out the steady state terms, you end up with a single difference equation in terms of the perturbation in the phase. And this perturbation in the phase is either going to grow or it's going to shrink. And this quantity here, which is written in terms of the slopes of the phase response curve. So this is 1 minus the slope of the phase resetting at the time neuron 2 receives an input, and this is 1 minus the slope of the phase resetting curve at the time neuron 1 receives an input. So the slopes are going to be the important thing to focus on. And this quantity has to be, if it's greater than 1, called the eigenvalue. If it's greater than 1, then the perturbation is going to blow up and the fixed point's unstable. If the quantity is less than 1, the perturbation is going to go to 0 and the fixed point is going to be stable. And so we can determine these stability criteria, these eigenvalues, for a firing mode with unequal time lags, for antiphase mode in which the time lags are exactly equal, and for synchrony. And right now I'm just talking about networks of 2 neurons as I illustrated in the previous slide. But you know, if math isn't your thing, the one take home is, in this sign convention, small positive PRC slopes are stabilizing, negative PRC slopes are destabilizing. And that's because when you take 1 minus a negative number, you have a number that's bigger than 1. So that's destabilizing. All right. So I said I was going to analyze networks of neurons and determine when they form clusters. Well, I'm going to make a very strong simplifying assumption, and I'm going to collapse an n neuron network that's connected all to all into 2 neurons. And I'm going to take one neuron, and I'm going to make it self-connected, and the self-connection represents the input from all the other neurons in its own cluster. And then the other cluster, it's going to have its own self-connection, and then the inputs, all the inputs between them are going to be collapsed, so you just get one big input from each cluster, and they're scaled by the number of neurons in each cluster. And this can easily be adapted to sparse connectivity, because you can just multiply this, and this is a scale factor. So we're saying that each individual synapse is now n over 2 times as strong, because you got n over 2 with n the total number of neurons in the network connections. So we're going to collapse everything down. And this is going to allow us to do a phase resetting curve on a cluster or on a stand-in for a cluster. This is actually a fairly common thing to do, to have a self-connected neuron stand-in for a cluster. And so now we redo the phase resetting analysis, and it's not really that different, except for that the neuron that we are performing the phase resetting analysis on has a self-connection. So every time it fires, it gives itself an input. And this internal delay is a parameter. So as we change the internal delay, as we change the conduction delays in the network, that's going to change the phase resetting curve. And it's also parameterized by the number of the neurons in each cluster, because that just gives you that scales the strength of the perturbation. So this is from the opposite cluster, this is from the same cluster. And again, this is the waveform of the spike, and this is what the phase resetting curve for an inhibitory input and a very strong inhibitory input, I might add, looks like. Right, and so these are just a couple of examples. On the left is from model, on the right is from experiments in John White's lab, specifically on PD positive, fast spiking parvalbumin positive basket cells in the entorhinal cortex. And in John White's lab, they self-connected a neuron using the dynamic clamp and then performed the phase resetting analysis. And Ruben Tkici-Hambrian in my lab did the same thing in an Izhikevich resonator neuron, which is a model. And in both cases, for strong inhibitory input, you get this sort of quasi-linear phase resetting curve with a discontinuity. And this discontinuity is going to be very important, and we're going to find it in the results that I'm going to show you today as well. All right, so this is the first results slide. And this is the results that Ruben Tkici-Hambrian in my lab got when he connected 300 Hodgkin-Huxley model inhibitory interneurons with all-to-all coupling and these bi-exponential inhibitory connections. But he varied the delay, the conduction delay, and he varied the normalized synaptic conductance. And it turns out that at low values of conduction delay and weak conductances, you get a two-cluster mode. But as these delays get longer, and the conductances get stronger, you get global synchrony. One other wrinkle is that in this region where you have the two-cluster mode, you also have global synchrony. It's bistable, but the two clusters are dominant. And we're going to try and explain all these observations using the phase resetting curve. So I'm going back to these maps, interval maps. And just to make the point that when you add delays into the system, you know in a synchronous mode, if everybody's firing in synchrony, then everyone is getting an input a little bit later, and that's determined by the conduction delay. And you need to, in order to get the phase, you need to normalize the conduction delay by the intrinsic period. So we know exactly in a synchronous mode at what phase the input's being received. And we know the stability criterion. All right, and then this is an anti-phase mode, and not to belabor the math, I just want to know that you can write a simple algebraic expression without and with delays. This is a line, 2 times the phase minus 1 plus 2 times the normalized delay. And if this line here, if it crosses the phase resetting curve, then you have a fixed point. And that you're going to be able to determine the stability of the fixed point by the slope at the point at which it crosses. All right.
Carmen, one question from Scott. Sure. Scott, do you want to ask yourself? Can you unmute yourself or?
Yeah, so just really quickly on when you were showing the 300 Hodgkin-Huxley network, so that parameter space, there's always synchrony in either a 1 or 2 cluster form. There's no asynchronous regime.
We did not find one. Now there's no noise here.
Okay.
Okay.
Awesome.
But we will return to that in just a bit.
All right.
Thank you for that clarification. All right. So here's the phase. This repeats the bifurcation diagram, and I'm focusing on the 1D slice and the 2D bifurcation diagram, and I'm particularly focusing on why do you get 2 clusters here at 2 milliseconds delay, and you do not get them here at 3 milliseconds delay. And so we performed the phase resetting analysis in this assumption that we can reduce the network to only 2 neurons, and we got these discontinuous, monotonically increasing phase resetting curves with a little discontinuity at the end. And this is common because this is a bi-exponential conductance, and if it starts just before the spike happens, it might not be able to stop it. But if it's strong enough, it will. But in this case, this is weak coupling. So we get this discontinuity because if you apply it soon enough, you can stop the spike from happening, and you delay it for a long time. But if you slip and you don't prevent the spike, then it happens and nothing much changes. All right, so this discontinuity is going to be really important. I'm going to talk about it a lot. It's a virtual negative, it's a discontinuity, but it functions as a region of virtual negative slope, which is highly destabilizing. So if you have one neuron receiving an input at a phase at, just on one side of discontinuity, and the other neuron is on the other side of discontinuity, when they receive the same input, it's going to have a vastly different effect, and that's highly destabilizing. All right, so we see that synchrony is stable because we know that at what phase synchrony you get an input. You get an input at the normalized delay. And these little crosses, so you've got a small positive slope, so synchrony is stable. And that's why we have synchrony along this line. But why do we lose the 2 cluster solution? Well, I previously said you can write an algebraic expression for a line, and when it intersects with the phase resetting curve, that gives you the phase at which an input is received in the antiphase mode. So this phase is received pretty late. It's pretty strong phase resetting. But as the line depends on the delay. So as you increase the delay, the intersection now falls in the discontinuity. So antiphase does not exist, and there is no fixed point. So this is why you can tell from the phase resetting analysis when it's going to disappear. Technically, it loses existence. And the antiphase is stable and synchrony is stable because we've got these small positive slopes. This one's not stable, but just because it doesn't exist. This is not, it's in this virtual region of very, very negative slope. All right, so there's another mode. I talked about synchrony and I talked about antiphase, but you can have an alternating mode in which the time lags are different. And in this case, you have to write a curve because there's two phases. There's a phase at which one neuron receives an input. There's a phase at which the other neuron receives an input. And so now you write a curve. And the intersection, this is going to intersect, I'll show it. It's going to intersect in two places. This is the curve. It's going to intersect at the phase corresponding to the short time lag and the phase corresponding to the long time lag. So it's going to intersect in two places. But remember, there was two stable modes. There's synchrony and there's antiphase. What separates these is this unequal time lag mode, and it is unstable or technically doesn't exist. It's a strongly repelling, but one of the points falls in this discontinuity. So technically the mode doesn't exist, but it does function as a virtual or ghost repeller that separates synchrony and antiphase. And so I need to explain what this little 1D bifurcation diagram is, and it's going along this line as you increase the delay. And so what the X's represent synchrony. So look over here at synchrony, we're going to call one of the time lags to be 0 and the other time lag to be 20. The time lags can be derived from the phases using algebra, which I could have shown on the previous slide, but it's too tedious. But they can't. The time lags can be derived from the phase resetting information and the fixed points of the map. And so synchrony, you have one time lag 0, and one is about the intrinsic period. Now, antiphase, the two time lags are the same, so they map onto each other. So we can calculate, from this information, where do these time lags lie? And then we can also calculate what are the time lags for the unequal mode. So for a 1 millisecond delay, they're 2 and 18. Time lag of 2, time lag of 18. So this tells me that if I take synchrony and I perturb one of those spikes by more than 2 milliseconds, that's going to make the synchronous mode go to the antiphase mode. And in this space of time lags, the 2 cluster mode is dominant. You only have a little bit of possible lags that go to the synchronous mode. So that's why the 2 cluster mode is dominant here. But at about 3 milliseconds delay, the unequal time lag modes, the clusters are all equal. We're assuming the clusters have equal numbers at this point. But the unequal time lags coalesces into the antiphase mode and disappears. So they cease to exist, and you're left with only global synchrony. So why does a stronger conductance cause the 2 cluster mode to go away as we make the conductance stronger? Now we're going to look at this slice, which is for stronger conductance. Now technically with no delay, there is a little bit of there's a antiphase prediction. However, if you get any delay at all, then you move this line that tells you where the antiphase mode would be into this continuity, and it's destroyed. And why is that? Because when you strengthen the conductance, you make the PRC steeper. And so that makes this line miss it. If it was less steep, it would intersect it. Steeper, it misses it. And basically, so the phase resetting analysis predicts that there's not going to be any, I've labeled them as, this is the antiphase mode, but it's actually, I'm saying it's unstable, but it actually doesn't exist. So it doesn't exist in its repeller. And however, synchrony is stable at all these delays. So we're able to predict these one-dimensional slices of the, and explain why you get synchrony and why you get 2 cluster. But what happens, and this is pertinent to Scott's question, what happens when you go to a sparsely connected network? Now there is no noise in this model, However, when you go to sparse connectivity, it introduces frozen noise. So in all-to-all, there's no jitter in either the 2-cluster or global. I'm going to skip over this in the interest of time, but when you have sparse random connections, that introduces jitter both in the globally synchronous mode and in the 2-cluster mode. And that's because the network doesn't look the same to anybody anymore, because you've got a binomial distribution in the number of synaptic connections because you flipped a coin and wiring up the network. So this introduces jitter. That's well known. And introducing jitter now has introduced a large region of asynchrony, where you don't get either the 2-cluster or the 1-cluster mode. And so how do we explain this? Well, this is the rasters for global synchrony at a delay of 3 milliseconds. And your jitter is on the order of 2 milliseconds. But since the delay is 3 milliseconds, you can tolerate that. And why is that? It's because your operating point is moved away from this region of unstable, destabilizing slope. But if your delay is too small and the jitter is bigger than the delay, then you're going to sample it and it's going to destabilize synchrony. And that's what happens for delays less than 1 millisecond, that the jitter is too big. But as you increase the delays, then you can compensate for the effect of jitter and keep synchrony stable. So, and then I just want to make one more brief point before moving to the next part of the talk. And that is that the 2-cluster mode is actually less destabilized than synchrony. So it's not destabilized. It's only destabilized in a small region. So the 2-cluster mode remains stable here despite the jitter, and it actually expands a little bit compared to synchrony. And I don't want to belabor this point. I only want to say that we have done the math and published it. Previously, we ignored within cluster synchrony, and we just assumed that the clusters were synchronized, but that's not necessarily the case. And the antiphase coupling. So this is within cluster synchrony terms, and you perturb one neuron out of that cluster and leave the other cluster intact. And the point is that having this antiphase coupling can enforce synchrony on a cluster that might not necessarily have synchronized on its own. And that's because when you look at the eigenvalues corresponding to the tween cluster and within cluster terms, you actually get a scaling term in this analysis. And you scale the eigenvalue that tells you whether you get within cluster synchrony, and you scale it by the 1 minus the slope of the phase resetting curve at the time when it receives an antiphase input. So that said, I know I'm not able to explain this in detail, but I just wanted to point out that there is a stabilizing influence of being partnered with an anaphase cluster on within cluster synchrony. And another brief point, I only talked about equal clusters. There many times we get clusters of unequal number as solutions, but you can show that if you have 2 unequal clusters, that one of those clusters is always going to fall into the discontinuity at a sooner than the equal cluster. So it will take the appropriate thing to look at is when does the 2-cluster solution lose stability, because everything else is going to lose, and stability is again a loose term. It's actually losing existence. But so it's appropriate to look at the equal 2 clusters, and you can ignore the unequal 2 clusters in this analysis. So this is the first part of the talk. And just to summarize, strong inhibition gives monotonically increasing delays in the PRC, and this discontinuity that often occurs in these inhibitory PRCs. If it's monotonic increasing, it's going to have a discontinuity. And it's destabilizing. Delays can avoid it, jitter can sample it, synchrony and 2-cluster can coexist in the boundaries given by the unequal time lags mode. Increasing delays or conductances destroy 2-cluster solution by pushing it into the discontinuity. Mutual anaphase inputs can stabilize a 2-cluster solution. Unequal 2 clusters lose stability, and really more accurately are pushed into discontinuity before equal clusters. And I didn't cover this, but we have also published that n-cluster solutions in general lose stability before the 2-cluster does, or are pushed into discontinuity. All right, so I'm going to now.
So Carmen, just to interrupt,
yes.
There's a question here from Alexandra. Do you want to go ahead, Alexandra?
Sure. So that should be a simple question, just to see if I've understood this correctly. So given the right delay and the appropriate conductance, is it that the 2 clusters occur because the cells from the 1 cluster mute the cells from the other cluster and vice versa? Is it that because of the PRCs that you showed,
Wait,
is it that the input
is the question on this slide?
So this is the anti-phase coupling. So I suppose it would apply to this slide. Yes. So I'm asking what happens when, right, so that when there's anti-phase coupling. So when, is it that the input from the one cluster, as you showed in the PRC, delays the spikes in the other cluster, and that's why the two populations fire in antiphase, sort of?
Wait, yeah, I'm afraid I'm not, which, do you know, is this the, are you asking how the antiphase cluster stabilizes the other cluster, or is it a totally different question?
That as well. I'm asking whether the cells from the one cluster, I'm asking whether the cells from the one cluster are actually muting the cells in the other cluster because of the PRC that you showed.
Are you saying muting?
Yes. So are they delaying
I think
the spike?
it's a terminology thing. Yes, I think it's kind of resetting it to a constant place. It's sending them all to the same place in the state space.
So based on the phase response curves
yes,
that you showed,
it's a very strong input. And I didn't mention this, but if you have a linear PRC or a quasi-linear PRC, basically that means that you're getting the same, you're getting the same resetting. The spike is going to occur. I'm flubbing this, but yes, I think muting is, if I understand you right, it's sending it to a place where it's going to be quiet and then take a while to recover.
I suppose, yeah, delaying it maybe is the best.
Yes. It was a terminology misunderstanding. Sorry. But all right, so to go to the second part of the talk, I'm going to talk about type 1 and type 2 excitability in the time remaining. And this was defined by Hodgkin in 1948. And this is from Izhikevic's book, that the point is that type 1 excitability means that as the depolarizing current is increased, a type 1 neuron can fire arbitrarily slowly, but a type 2 neuron has got a lower bound below which it cannot sustain frequency. And so as you depolarize the neuron, then it will spike, but it will die out because it can't maintain spiking at a slower frequency. So that's type 2. And why is that? And this is an explanation in terms of the steady state IV curve of a neuron. And so in general, a type 1 is going to have a negative slope conductance region, and type 2 does not need one. And the reason is at steady state in type 1, you have a net inward current. So you can spike really slowly because it's always inward. There's always some inward current there. And these are the instantaneous curves, which I'm not going to talk about. But in the type 2, when you go to steady state, if you stay there long enough, the net current in the interspike interval is outward. So at short, for short times as you depolarize, you have inward current. But if you stay in that region, in that interspike interval too long, then the outward currents catch up and shut you down. So that's why type 2 neurons have to fire faster. They can't fire at all because the outward currents will catch up and shut down the firing. Whereas that's not a constraint for type 1. Why do I care? Well, this work with John White, actually this is work from John White's lab. It showed that parvalbumin positive fast spiking basket cells, which are the neurons, and I didn't mention this, but these are the neurons that we think are most closely implicated in gamma oscillations. And he showed that they are likely type 2, whereas some work that Francis was involved in is in agreement with the traditional thinking dating back to Wang and Bazaki 96, if not longer, that these parvalbumin positive fast spiking basket cells are actually type 1 and can fire arbitrarily slowly. But I would like to, Lauren Hewitt in Darren Brager's lab, and I think Darren's on the call, very kindly shared some data with me. And I only picked the examples that, that speak to my point, of course. But so these are actually from CA1 neurons, fast spiking neurons in area CA1. And she did some frequency FI curve on them, and gets kind of a classic response on this cell, and actually sees stuttering on this one. So I'm just wondering if maybe there's heterogeneity in cell type, or but anyway. So there's also additional evidence for type 2 in these MEC parvalbumin positive cells from John White's lab, and that's that they resonate. If you give them a little inhibition, they ring, like an Isaac-Head's neuron, they ring a little bit. And this is what you would expect from the circular movement around a subcritical hop bifurcation. And they can even emit, a rebound spike due to a brief perturbation. Now, this is not like something engaging IH or something like that. It's a brief perturbation. And so we published this same paper, we published the suggestion that cycle skipping, and I'm going to define that in the next slide, but that cycle skipping enables you to synchronize by firing only when your spike is lined up with the population, and being silent when it's not, being muted, for example, when it's not. And so we suggested that cycle skipping was much more robust in type 2 networks, but we used different models in different parameter regimes, and we wanted to revisit this issue because we felt that that wasn't a fair comparison. And so there is some evidence for cycle skipping in an in vitro model of gamma from Hajos et al. So this is in dentate gyrus, I believe. And so these are cell attached recordings showing spiking in an individual neuron, interneuron, and this is the field LFP, the field potential. And the neurons are fairly tightly locked. And in fact, you can see a peak in the interspike interval, and you can maybe see a peak at the harmonic of the basic frequency, because they fire on average every other cycle. And then here's another example of cycle skipping. These are two mutually coupled in a slice by Hu et al., two mutually coupled fast spiking cells. And they depolarize one, and then they depolarize the other one. And this is in a slice, so they have control over it. They're silent normally. And they depolarize them, and now this one neuron will stop the other one from firing unless its firing is precisely aligned with it. So it skips some cycles and fires on others. So this is what I'm calling cycle skipping. And it's also observed in a model by Economo and White. This is a model of gamma. And the excitatory cells, they fire with an exponentially distribution for the E cells, but they have a multimodal Gaussian distribution for the I cells, because the I cells are skipping. And in this model, the I cells happen to be type 2, and they happen to be connected by shunting inhibition. All right, so in our previous work—
Sorry, Carmen, just a quick question.
Yes.
This is Francis, sorry. So in that previous slide, where you're showing the previous one to this, when you were showing, those fast spiking the coupling was a dynamic coupling, or was that—
No. They found that, thank you for asking this, I should have made it clear. They identified cells that were reciprocally coupled. So it's the natural synapses.
Okay, and it's monosynaptic.
It's not, that's why I was so impressed with it. It's not artificial.
Okay, and they know it's monosynaptic also. Monosynaptic.
Yes, well, they did I can send you the paper.
Yeah. Okay, that's fine. Thanks.
All right. So anyway, there's cycle skipping in this model, and our previous work only looked at hyperpolarizing inhibition, meaning that you get an increase in conductance, but you also get a deflection in membrane potential. Whereas in shunning inhibition, you get an increase in inhibitory conductance, but you may not get much of a change in the membrane potential. And so we wanted to look at both of these cases, and we wanted to do a fair comparison in a model that this is the Hodgkin-Huxley a reduction that John Rinzel pioneered and Ruben Tukiji-Hambrian adjusted, so that the only difference between the models, and this is a 2-variable model, it's got membrane potential and it's got N, which is the activation of the potassium current, and it's yoked to the H variable in the Hodgkin-Huxley model, and M, which is the activation of the sodium current, is made instantaneous. So it's a 2-dimensional reduction of the standard Hodgkin-Huxley model. And Ruben calibrated it so the spike shape was the same, the FI curves were to the degree possible the same, and there's heterogeneity in this model. So the intrinsic frequency of these neurons is from 42 to 62 in both cases, and their resting potential is, their input resistance is the same, and their time constant is calibrated to be the same as well. So we wanted to change absolutely nothing except the excitability type, and then make a fair comparison on their synchronization properties. And so this is a fast-slow analysis. It's a phase plane. This is the potential nullcline, membrane potential nullcline. This is the activation curve for the N variable, from the Hodgkin-Huxley N variable. And and the intersection, of course, of the two nullclines gives you the fixed points. The thing about the fast-slow analysis is the slope of the membrane potential. If it's the fast variable, the slope of this null line gives you the stability of the fixed point under fast slow, that if n is very slow compared to v. And so we have the slope here is negative, and that gives you, and don't confuse this with my previous talk on PRCs. We're talking about the slope of the potential null line. If it's negative, that means it's stable. If it's positive, it's unstable. So we have, and in type 2, it has at rest, you have one stable fixed point. There's actually 3, but in type 1, but the other one's far away, and I'm focusing on the 2 that are going to coalesce and disappear in a saddle node on an invariant limits on an invariant circle bifurcation. So this is at rest. This is as you increase, you depolarize the cell, and this is the SNC when the slow and the fast coalesce, and they actually have to touch the limit cycle at that point as well. And then for a hop, it's a change. It changes from the negative slope to the positive slope region of the potential null line, and that's what gives you. So it's a hop, and now this becomes an unstable fixed point. So now you have oscillations. So this is just to show that the model is calibrated to have a type 1 and a type 2 that are as much alike as we could possibly make them, so we can examine them in the same parameter regimes and compare them head to head. And if we used a 300 single compartment reduced Hodgkin-Huxley models in neuron by exponential inhibitory synapses, now we have uniformly distributed delays and no spatial structure. Heterogeneity and the applied current, as I described before, random sparse connectivity, and in this case, we do have independent Gaussian current noise. So there's noise in this iteration. And we actually found that there was, well, one finding is that initially the coupled oscillator model, meaning the mean regime model of gamma oscillations being mediated by coupled interneurons, was criticized because it wasn't very robust to heterogeneity, and specifically there could be suppression. So some neurons would be suppressed. And what we found that, you know, they both do cycle skipping, and these are these multimodal Gaussians at harmonics of the network frequency due to skipping one or more cycles. And we found that they cycle skip very robustly, both types do. So we didn't actually find that type 2 cycle skip more robustly. What we did find is that it was less prone to suppression. So the average participation, there's less suppression as compared to type 1. And you can see that here, that type 1 has and the slower neurons are at the bottom. So they're more suppressed in type 1. And again, the similarity in performance is type 1 and type 2. The red shows the tightness of the synchronization, the vector strength of the spikes that are emitted. Like I said, many are skipped. And the participation, there's not a huge difference. The CV of the participation and the amount of suppression was the only place where we really found a difference between type 1 and type 2. And I should remind you that I'm dealing with hyperpolarizing inhibition at this point. And I'm going to show a little movie, maybe. Yes. All right, so in this movie, this is the type 1 with hyperpolarizing inhibition. You see that, okay, this is the average null lines across the heterogeneous population. And so they wax and wane. The memory potential null line waxes and wanes with inhibition. So that's the minimum inhibition point. And there's a slow channel opened up between the null lines, and the neurons can escape. The red neurons have the fastest frequencies. The blue neurons have the slowest frequencies. And you see the blue neurons don't escape very often. And that's because in this slow channel, they tend to line up according to their fixed point. And the blue neurons have leftward fixed points, and the red neurons have rightward fixed points because they have more bias current. And they line up along here, and the red ones have a better chance to escape. And the reason they can fire arbitrarily slowly is this slow channel that opens up okay, there's a fixed point, now the fixed point goes away, and there's a slow channel here. And anyway, so I will try and summarize this on the next slide. That with minimal inhibition, you get these neurons lining up in the slow channel, and the fast ones have an advantage because they're lined up according to their their intrinsic firing frequency. And then accumulating inhibition creates this fixed point, and that causes the silence between cycles. And then, well, here, this fixed point is at maximal inhibition. And the fixed point's just basically moving in the fast direction. So it's really amenable to suppression. It's not really given a fair playing ground to everybody. But the picture is different for hyperpolarizing inhibition. And that's because now we don't have this slow channel anymore. The neurons don't line up. And there's actually the black dash line is the quasi-threshold of Fitzhugh, and it collapses into the unstable fixed point that unstable limit cycle that surrounds the stable fixed point in the bistability near a subcritical. And so, but basically all neurons have kind of a fair and equal chance at escaping, because they kind of fall, the threshold is kind of in a horizontal as opposed to a vertical direction. And so they escape and they're resistant to suppression. And this kind of summarizes the previous movie. But in the interest, I want to move on because I don't really have very much time left, and I want to get through a little bit more of it. And I just want to say that for shunning inhibition, type 2 loses its advantage in homogenizing participation. So shunning inhibition, type 2 no longer has an advantage in participation. Everybody's still cycle skipping robustly. Again, there's no great difference in the ability to synchronize in the participation, but actually type 2 is a little bit more suppressed for shunning inhibition until the noise becomes too strong. Yeah, I want to back up. I'm going to come back to these multimodal interspike interval histograms. So I just want to make sure that, these correspond to cycle skipping. And if you're not skipping cycles, then you would just have one Gaussian peak. All right, so this is the network type 1 with shunning inhibition. Okay, I'm having trouble with my movie. All right, there's my movie. Okay. All right, so this is with shunning inhibition, and you see that we're gonna see some blue ones escape. Yeah, so it's not. They're not lined up because the shunning inhibition doesn't string them out like the hyperpolarizing does. It kind of makes them more compact. And so there's no longer that tendency for the faster neurons. There's no longer such a strong tendency for the faster neurons to win and suppress the slower ones. And then there's the final interesting point about these phase plane analysis is for shunning inhibition, the quasi-threshold, the black line here, moving black line, is going to collapse into a unstable limit cycle, and it's going to trap neurons in there. And so that actually contributes to suppression with shunning inhibition. So just to try to summarize, for shunning inhibition, you don't really see this lining up along the fixed point that you did with the hyperpolarizing inhibition that was stringing them out in a line. And there's a bit more, and for some reason, there's a bit more motion in the slow direction, which is also going to tend to shake things up as opposed to confining it all in the fast direction. And then this unstable limit cycle, we blow this up, it tends to trap type 2 neurons. So the bottom line is that shunning inhibition is a little bit better for participation for type 1, they participate more, and hyperpolarizing inhibition is better for type 2, they participate more. And I'm going to skip through here. I've just got a couple of quick little points to make, and that's that we're going to now look at theta nested gamma. So theta power is highest when it's driven, gamma power is highest when it's nested in theta. So we're going to look at some theta drive, and we're going to sinusoidally modulate these type 1 networks versus type 2 networks. And they are recruited, it's similar to our previous results, we're able to recruit type 2 neurons more strongly. The gamma oscillations have higher phase amplitude coupling with the theta than the type 1, and this, the gamma power is just, the gamma amplitude is larger on the bottom for type 1 than at the top for the bottom for type 2 than the top for type 1. And this is just a summary. So if basically we're looking at the phase amplitude coupling of gamma amplitude to theta phase, and when we look at the difference between the two cases across a space of modulation frequency and modulation strength, or depth, I suppose you would call it. And in general, the red means that type 2 has got stronger, it's evoking a stronger gamma coupling, stronger gamma amplitude relative to the theta phase. It's coupling very tightly more tightly to the gamma phase, to the theta phase. Sorry, I'm struggling to get these last few points in.
Yeah, sorry, Carmen. Just very quick question, just so that yeah. So your LFP that you're simulated.
Yes, that's just the sum of the synaptic currents. Some of the synaptic conductances. Sorry, I'd have to actually Ruben did it. I don't remember. I guess it would make more sense to do the currents.
Right. Okay, so is it the currents or the conductances?
Well, I don't think Ruben's on the call.
Yes, he is. Actually, I can see him here.
Well, can he answer that question?
Can you answer that question, Ruben? I see his name, but.
All right. Well, I'll have to come back to that.
Yeah, we'll come back. Yeah. Okay.
I think the current makes more sense to me, but I don't know what was actually done.
Okay. All right.
I don't remember.
Yeah. Oh, Alexandra,
yeah. So on that note, the coupling, is it defined as the modulation index between the gamma and the theta?
It's the phase amplitude coupling. So it's the vector strength. Let's see. It's the amplitude. Aha. So he did a Hilbert transform to get the amplitude of the gamma, and then we know the theta phase. So it's the coupling between that amplitude and the theta phase in terms of the vector strength. And normally, sorry?
Both gamma and theta LFPs.
Well, it's gamma amplitude coupled to theta phase.
And both gamma and theta LFPs were calculated by the conductances. Both frequencies, right?
We don't have to. Let's see. This is the inhibitory. This is just a drive. We just gave it a theta drive, so this is constant.
The theta drive is not generated by the population. It's given. It's
yes. It's just driven. And this amplitude. That is a good point. Yeah, I
see Ruben here. He's unmuted. Could you?
Yes. Answer. Is it conductance or?
Ruben, can you hear us?
Oh well, I'll have to get back to you on that.
Okay. All right. Okay. So why don't you wrap up, and so then we could continue questions. Yeah.
Well, skipping to the conclusion. I'm just going to skip to the conclusions because there's one point I want to make real quickly. But anyway, so basically, there's at least some parvalbumin positive fast spiking interneurons, which are important for gamma and gamma theta coupling are type 2. So I questioned the implications, but for synchrony. But it turns out cycle skipping and theta nesting emerge naturally over a similar range of parameters for both type 1 and type 2. And they produce multimodal ISI distributions. And as would the clustering I discussed earlier. Type 2 neurons are somewhat more resistant to suppression for hyperpolarizing, whereas type 1 are somewhat more resistant for shunting. And what it finished totally was that phase amplitude coupling of theta nested gamma is somewhat stronger for type 2 networks with hyperpolarizing inhibition, and consistent with the results on suppression, it's somewhat stronger for type 1 with shunting inhibition. So maybe there's like some relationship. You could think maybe there could be some coevolution where if you have shunting inhibition, you might want to go with type 1, and if you have hyperpolarizing, you might want to go with type 2. All right, but I had a hard time getting this paper published. It's published in eNeuro, but I was criticized for the multimodal Gaussian ISI histograms. I was told nobody in neuroscience is interested in coupled oscillators because everybody knows that the brain is always not in a mean-driven state, but in a fluctuation-driven state. And so it's not, the coupled oscillator theory is not relevant. I didn't buy that, but I was told that. And so I would just make some brief, do we care about the individual oscillatory properties of individual interneurons? Because there is this competing hypothesis that there's a stochastic population oscillator. The individual neuron properties don't matter. They're in the fluctuation-driven regime as opposed to the mean-driven where they oscillate. Neurons fire very sparsely in this mechanism, and a random walk to the threshold based on their random inputs produces it's basically a random walk, and so the interspike interval distribution are the threshold crossings of a random walk, of a diffusion process. And this dates to Brunel and his colleagues, but it requires very, very low firing rates for the neurons, and it produces an exponential ISI, and it produces an oscillation in the LFP. But it requires low firings. And Guillem Villa, who's on the call and is in my lab now, has reproduced this in a network of 500 sparsely connected leaky integrating fire neurons. And with tiny delays, they're asynchronous and they have an exponential distribution. With a 2 millisecond delay, you get a population oscillation in the firing rates. You still maintain this exponential distribution, but the average firing rate for the individual neurons is less than 4 hertz, and 150 hertz is the population oscillation. So I'm going to just throw out some data that was very kindly allowed me to show from Margaret Donahue and Laura Colgan's lab at UT. And it's just one example, but it's from a putative interneuron in a freely moving rodent during an episode of fast gamma. And Laura defines fast gamma as 65 to 90 hertz. And the individual neuron this is the histogram, and it's firing at about 90 hertz is the first peak of and its firing rate is fit by a multimodal Gaussian. And here it's a bimodal Gaussian, and there's not that much area in the second peak. But this would indicate that it's firing on almost every cycle and skipping sometimes a cycle. So and it doesn't fit an exponential area. But my take home point is I think we need to look closely. What is the data from interneurons during oscillations? And so this is only, maybe they do have exponential distributions when we're not doing gamma oscillations, but I think it's still an open question whether these interneurons are acting as coupled oscillators or if we're always talking about stochastic population oscillator. And I want to thank all my collaborators and stop here. And the ones that are bolded contributed to the work that I presented today.
Okay, thank you very much. I'll clap on behalf of everyone since we have this virtual. Okay, and so just looking at the time here, yeah. So we'll take some time for some questions. If people gotta go, you gotta go.
And thinking about it, it had to be the conductance. Otherwise, it would have been all over the place.
Yeah, it's Ruben.
Okay,
Yeah, I apologize. I didn't have a Zoom on my system, so I'm a little bit unfamiliar with this. Yes, it's a conductance. It's a conductance, but we assume that it's not just conductance in the interneurons. It's conductance with long delays, long time constant, which is in the pyramidal cells.
Yes, that's an important point I remembered. Of course, the LFP is thought to be the inputs that the pyramidal cells get.
Yep.
So and there's a different time constant of decay. So yes, thank you for bringing that up, Ruben. I'd completely forgotten, and it's important. It is an important detail.
Okay, thank you. Does that, Alexandra? Is that?
Yes, I have 2 more points to ask about if no one else wants to ask a question.
Yeah, go ahead. Scott has his hand up and he'll go next. Yeah.
Okay, so first, regarding your last point about whether or not the cells are intrinsically firing periodically, that's something you said we don't know, we'll have to look into further. I'm just thinking of some data from Williams' lab, where I think they've showed that the pyramidal cells fire sparsely, not in every theta cycle.
I agree that the pyramidal neurons fire sparsely.
But we don't know about the interneurons, is what you're saying, right?
Yeah, I don't think it's been shown that at all times that they're in a balanced state and in a fluctuation-driven state. I think that we should look, during episodes of fast and slow gamma.
Right.
I think we need to look at that, and
I guess the same lab has shown, well, again, from few recordings, obviously we need hundreds of cells to have like a good sample, but from the few cells that I've looked at, it seems that the parvalbumin cells fire at every cycle, but it's the OLM cells that can also fire very stochastically.
Wait, now, which?
The parvalbumin, the PV.
Oh, no, but which lab?
Oh, Williams' lab, Sylvan Williams' lab.
Oh, yes, yes, and actually, I have some data that, I've been looking at some data from a paper that Sylvan Williams published with John White.
Right.
Felix was the first. I'm blanking on the first author, but it was on the intact hippocampal preparation.
Mm-hmm.
And I've been looking at the data from those interneurons, and some of them are firing at ripple.
Oh, that's very interesting. Okay,
Yeah.
so
And I hope I don't know if John or John White authorized me to talk about the data. So.
Well, I mean, I guess what I would just throw in there is like KT's original, like 2013 model with this fast spiking PV cells, Alexander, that you know about from that prep are these very, like the 90 hertz, like very high frequency also.
Right.
Yeah.
I was thinking of Carey's paper, obviously, right, Francis?
Yeah.
So where they showed the OLM don't fire at every cycle, but the PV basket cells do fire at every cycle.
The basket, I need to see that paper.
Yes.
That's
so
why you get you to come and talk. Okay.
It's Gary Ha, so H-U-H.
Yeah, you add it in the chat.
Sure.
I would have liked to have had that paper in my response to the reviewers.
Yeah, it would be useful. So yeah, I'm gonna write it on the chat. I have one more question. So if I understood correctly about the theta rhythm imposed in the model, that is not generated by the pyramidal cell. It's externally.
Well, we were looking. I mean, I think that there may be some internal theta generation, but we were specifically thinking of theta drive from the septum, from the inhibitory neurons and the bursting.
Right, so it's an in vivo type situation. I was wondering whether you were interested to see, well, if the network is generated, the theta, what would happen with the coupling that you showed, the theta gamma coupling. In that case, it's something that
we haven't moved to we don't have intrinsic models of theta generation yet.
Right. Yeah.
So,
okay.
Stay tuned.
All right, I'll just put the paper on the chat.
Yeah. So Scott, you had your hand up. Go ahead.
Yeah. So great talk, Carmen.
Thank you. I don't see the chat, but go ahead. Go ahead, Scott.
Yeah. So I loved how you brought up that sort of dichotomy in the literature about whether PV cells are type 1 or type 2. Do you think that we're just sort of using PV as a too broad of a classification, or do you think that this is actually indicative of someone being right and someone being wrong about their characteristics?
I mentioned there could be heterogeneity, but certainly now the basket cells from John White's lab, they do the I showed the staining, I forget what they call that, but post hoc, they stain the cell and they see what it looks like and where it is. So those were the ones from that paper. I'm pretty sure we're sure of that those were basket cells, and that they were PV positive, and they weren't. What are the other ones, like axoaxonal cells? Some of the other ones that are PV positive.
Ratified and axoaxonic. Yeah.
So then, if that's the case, have you started looking at all at sort of networks with mixed type 1, type 2 PRC profiles and sort of?
Ruben did, and that didn't make it into the paper. But yeah, he did, and what my recollection was that the type 1s, the mixed ones, behave more like type 1.
Okay.
Scott, so if you have a mixture and you have a hyperpolarization, so network behave as the weakest subpopulation. So if it's a hyperpolarization, the network behave as a type 1. If it's a shunting inhibition, its network behave as a type 2, as the weakest population in subpopulation.
Again, thanks. I did not remember that. Thanks, Ruben.
Awesome. Thank you.
So you know whether certain cell types behave more as type 1 versus type 2, like the axoaxonics that you mentioned before, relative to the PV basket or the bistratify?
I've been laser focused on PV parvalbumin positive fast spiking cells. I haven't really looked at other subtypes. But it's interesting because like Imre Vida, he said that he had data from dentate gyrus, but I can't find it published anywhere that showing this type 1 FI curve, for example. And and it's not trivial with noise and can you really tell if the threshold, some of these the ones in John's study, their minimum cutoff frequency was 80 hertz. So that seems clear. But if your cutoff frequency is 2 or 3 hertz, who would know?
Yeah. I mean, actually, one comment I could add here is, so you had mentioned Katie's paper, but the earlier paper of Ernest's, which was in collaboration with Marlena Bartos, that was, I don't know if you looked at that paper way back. And so this was dentate gyrus fast spiking cells. And according to Marlena, like she didn't really think, it mattered whether it was dentate gyrus CA1, CA3, or whatever. It's fast spiking. And there it's clearly, they did really small step sizes so they could kind of get down to see like that real base in terms of type 1, type 2. Mm-hmm. And so, yeah, it also seems like it kind of got quite slow. But then it sort of starts becoming, well, experimentally what's kind of going on, because, depending on how long your steps are, right? So if it goes really slow, right? I mean, one spike in 1 second versus one spike in 2 seconds, you know, then it becomes an experiment.
Right.
So
I only look at
yeah.
intervals. I don't count spikes. I only look at intervals.
Right, but you need the spikes for the intervals, right?
Right.
Yeah, yeah. So anyway, so it starts becoming a sort of experimentally technical thing. So I'm not sure if one could really.
Well, there's that alternate thing about whether they exhibit resonance to a small, when you
Right.
get that circular motion around the hop.
Yeah, yeah, yeah. That's something different. Yeah. Okay. Okay. Sorry. Let me just. Okay. I'm looking. I don't see any virtual hands, but if anybody wants to, or in the chat. So if there's a last chance before I end, for questions. As mentioned, this is taped. So, you know, if you go back and look at the slides, and then I'm going to volunteer Carmen, if people have questions subsequently, to contact you. Is that okay? Yeah, yeah. Because I mean, there's a lot of information, so we're trying to look at papers and come up with subsequent questions. Okay, so to just end and just express our gratitude for Carmen to, you know, meet virtually and give us our first online CMM KCN talk. And thank you very much. And stay safe and healthy, everyone.
Thanks.
Virtual clap. Okay, so what I'm going to do is I'm going to put everybody into the waiting room. So stay on, Carmen.
Sure.
And I'm going to put everybody into the waiting room, and then we could chat a bit more, unless anybody wants to have sort of more individual chats. Unless you say, okay, if you can see the chat, Carmen, there's all kinds of thank yous going on. So
I don't actually see the chats. Here we go.
Great talk. Long live, long live PRCs. Okay.
Yay.
All right. Okay. Okay. So I'm gonna put
I see them. Very nice.
Okay. So anybody else want to stay? I'm gonna just chat with Carmen myself, and unless you speak now, I'm gonna kick everybody out.
Well, is that paper in the chat, Alexandra?
Oh,
you
sorry.
muted.
Oh, I got, you have to, okay, let me unmute. I'm just going to unmute.
Yes, I mean, someone else actually put it on the chat. I think Francis did, yeah.
Yeah, I did, yeah. Okay, but I didn't give the whole link. It's Carrie Hewitt, all journalism.
Oh, I can do that, I can do that. Yeah. And I would like to stay, Francis, if I'm allowed, unless...
Yep, that's fine.
Yeah,
okay, so anybody else want to stay before I kick everybody out to the waiting room? And I'm okay, all right, so we'll just keep... So Alexandra, I'll just let you back in after, because I have a global way of doing this.
Sounds good.
I think. All right. Okay. Let's see. How do I do this? Okay. Allow flight to. Okay. Enable waiting room. Yes. Okay. And now. Okay. All right. Okay. Yeah. So okay. Actually, you know the way I could do it is I can actually put. Yeah. Oh, I see. Yeah. Okay. So now I could write. Okay. Yeah. All right. Okay. So I think, right. Yes. Okay. I see. Yeah. All right. I'm just putting everybody except Alexandra into the waiting room, because they didn't speak up, and so it's too late. Yeah. And I know you have. Okay. All right. Okay. Yeah. So I mean, I think I'm just starting to talk while I'm doing this. I'm multitasking. So Alexander, if you had particular questions, I'll just finish putting everybody into the waiting room here.
I'm trying to paste the paper.
Yeah. Did you put the DOI link? Can you get it?
Well,
Alex,
this is
yes, I have it. I could actually even show the particular cell profiles if like I could.
Oh, you mean like from the paper?
Yeah, and
right.
I was actually really devastated when I got those critiques. I emailed Charlie Wilson to.
So what actually could you expand on that? Was it like sort of this mean? So there's this sort of the fluctuation. Like
I don't think it was mean. I think it was someone who was very cortical centered, and they just thought coupled oscillators were of no interest. And so I got really upset, and I asked Charlie Wilson if he thought coupled oscillators were of interest, and he said, and particularly interneurons, and he said the basal ganglia is full of inhibitory interneurons that oscillate.
Yeah, I mean, so actually what I was trying to understand, like when you said
oscillators.
Yeah, because it's kind of, part of it is like how much of it is people's perspective on, you know, the sort of the mathematical side of things and the experimental side of things, right? And so you know, when I said mean, I meant mean,
Yes.
mean driven versus classification driven.
Yes.
And so that paper by Susan Schreiber, you know, where they talked about that. So that's a model, and you know, it's always this sort of thing about what the interpretation is and what one means by it, right? And so when those kind of criticisms come up, I like that you referred to, I think they're very unfair, and it's sort of like people just sort of hand wave and say, okay, well, it's not like this, like without talking enough specifics of what the details are. The details always matter, right?
Right. And I talked to Charlie, and he said, well, what about the basal ganglia? And in fact, he said, somebody told him once that there weren't any, he gave a talk and someone said, well, that doesn't happen in the central nervous system. And Charlie said, the basal ganglia is the central nervous system. I mean, some people are just very cortical centric.
Yeah, no, I mean, absolutely. I mean, like Alex just had a review that should come out in Frontiers with the VIP cell stuff. And he was trying to do this hippocampus cortex, you know, just the whole point about them, you know, these inhibitory cells that inhibit only other inhibitory cells. And the early work from Lisa Topolnik and that who's very aware of it because she's working at hippocampus was in hippocampus. So it was like Samarji and all these earlier people. But the reviewers, I didn't know who they were, but one was obviously a cortical person. And they said, oh, is there such a thing? I can't remember the details, but it's so true in hippocampus, but it's just that it hasn't been done in a clear way in cortex, right? And so there's this view, you know, it is what you know kind of thing, right? So it was just funny listening to Lisa's comment.
It really bothered me because it made me think, am I wasting my time? And so then I went and talked to Brent Doiron, and you know, and Brent's very cortical centered, and he said there's really no evidence that everything is in the balanced state, in the fluctuation driven state at all times.
Yeah. Well, you know, okay, this is my opinion. I mean, Jeremy's not here, but I've talked to him a bit about it too, because, you know, there's always a context associated with it, right? I mean, that's sort of like a theoretical nicety, so to speak, if I put it on that one, at least my understanding of it, like this balanced state, right? So that from which you can kind of have these sort of theoretical understandings and do analyses, right? But whether that's actually going on in the real biological system, you don't know, because it's sort of it's sort of set that way. So unless you kind of derive something from it, some context that could be taken back to be tested experimentally, it's hard to kind of get past that, right? And so, you know, I mean, so Jeremy, who's great, is very aware of this kind of stuff, right? And I think sometimes those assumptions, those underlying theoretical assumptions are hidden, so that it's like, well, what's really important, right? Whereas, I mean, so actually I want to talk to you a little bit more about this, specifically this type 1, type 2 business, is that, you know, the math is very precise, and that's great. And that's why, you know, at least that's why I've always done math, right? It's like black and white. It's clear what is going on, right? This bifurcation stuff. And the challenge is trying to kind of impose those insights into the biological system, right? And so my present thinking, and this is kind of wanted to ask you, Carmen, is that, actually now listening to your talk, I hadn't actually seen your most recent paper, the 2021. So I thought actually I want to go and look at it more closely, because it's really nice, because you guys really try to kind of keep, like, do a fair comparison, as you put it, right? Because that's always that sort of thing. Like, you know, it's set up in a certain way, and it depends on what you're looking at and things like that. So I want to look at that more closely. But my question to you in some sense is, based on my limited experience with sort of the experimental data and collaborators, the type 1, type 2 business becomes more gray, I would say, as you get into, you know, the details of the biological stuff you're looking at. At least this is how I feel like. So it's helpful to a certain extent. And then unless you can kind of impose the type 1, type 2 beyond the single cell, then it starts becoming, well, I don't know how to think about that type 1, type 2 in the real biological system. So just be more specific. The Ho et al. Paper, that was Ernest's paper way back, where we have Marlena Bartos's data in there with the fast spiking, and basically his thesis was called, if you want to be slow, you have to be fast. And basically it takes advantage of this sort of kink in the FI curve. I don't know if you remember that.
No.
Yeah, it's an older paper, but it's sort of, this is where sort of my thinking started to become what is type 1, type 2, like how much should or could we worry about, it's more how much could we worry about it as we get into the biological messiness, I guess is kind of really what I'm saying or wondering. And so that paper, he sort of showed like it was just a simplified Isakiewicz-like model also, but it's now trying to understand these sort of, you know, slow population oscillations, and this is actually Liang's data, Alexandra, not Sylvan, so this was earlier stages, but the idea is that you can get a slow population output just from purely fast-firing cells. And so the mechanism there that he proposed had to do with this kink. So you have this sort of biasability in the network. So it didn't have anything directly to do with type 1, type 2 kind of stuff, and it was more to do with the kink. So Marlena's student kind of did these really fine experiments to show that there is such a thing as a kink. So that's sort of different. But my point is, it's sort of, once you start dealing with whatever population, theta, gamma, in the particular biological system, then it starts getting tricky to how.
Right. Well, the directions I'm looking at is, let's see. So in the medial entorhinal cortex, Fernando Fernandez and John White's lab has shown that these that they depress. There's a lot of synaptic depression in the PV to PV and the PV to stellate cell synapses. So I'm wondering if that impacts the different mechanisms, stochastic versus coupled oscillator differently. And I'm also, there's a model of grid cells, Pastel et al. Of Matthew Nolan's lab. Mm-hmm. And I want to put these type 2 neurons in grid cell models and see if it makes any difference, and also put the depressive.
I mean, like if it makes a difference between the type 1 and the type 2.
Yeah.
Yeah. I mean, the way I was thinking based on just your talk right now, because I hadn't seen the very recent work, is that, so just so you know where I'm coming from, the way I kind of think about these is like, very much always want to take advantage of any of these theoretical insights, because the biology is so messy and complicated, right? And so it seems like what you've essentially shown is that maybe a way, the reason to focus on the type 1 or type 2-ness of whatever cell we're talking about is because now when you think of disease states like epilepsy, when you have shunting versus hyperpolarization inhibition, it matters. And so it's kind of like, so I'm kind of coming in it the other way, right? Like,
it mattered a lot less than I thought it would, though.
but didn't you say, okay, I have to, okay, so I saw that was a difference. Like, could you go back to your slide 1 for the conclusion of that part? Because it's sort of like, if you, so Alexander, you probably know about, that work, I can't remember who initiated it, but this whole thing about, I know Steve Prescott did later stuff, but different people, the type 1 to type 2 changed when you have the cholinergic imposition, right? But the important thing, or not important, what they were basing that on was theoretical work, I think from Bird, that it was only type 1 that can synchronize, but that was in purely excitatory networks. So there's always this underlying theoretical basis that kind of doesn't get said clearly. Right. So now there's kind of a disconnect, right?
So I wonder what we know about the hippocampus, the interneurons, whether they're type 1 or type 2, whether we have any biological basis to assume that they belong in one category or the other. I don't know if you know anything about that.
Well, we published on MEC, and the fast spiking parvalbumin basket cells in MEC have a cutoff frequency cutoff of about 80 hertz, and they have this resonance to a brief, you could get resonance to like IH or something, but for the spiking mechanism, it has to be like a brief input that you get ringing. And then we're also looking at CA3, and CA3 looks like it's got it also is type 2-ish. Mm-hmm. But, and then Francis has looked at type, and in fact, I in that paper, y'all did more fine steps to, I think, on some of them.
So CA3 type 2-ish and CA1.
Yeah, but then I did show data for, that's some very preliminary data from Darren. Okay. And Darren was on the call. I don't know if you know Darren Brake.
Yeah. No, I don't know him, but I see.
He was on the call.
Do you know him, Alexandra, or?
I don't think so.
Okay.
I don't know him either. I just found Laura Colgan as a friend, and I've got data from Laura, which, and I asked her if there was somebody that was doing in vitro work on these cells, and Darren is, so. She put
sorry, go ahead.
Yeah, that's maybe something that can be even assumed in a reverse engineering way. Like, which one of the two types you showed has stronger phase amplitude coupling? I don't remember that, like from your talk today.
For shunning inhibition, type 1 was better, and for hyperpolarizing inhibition, type 2 was better.
Right. So.
But I don't think it was huge.
So first, we need to look whether we have shunning inhibition or hyperpolarizing inhibition, and then also look at the two types. But okay, so it wasn't huge. I was wondering.
I actually felt like I had published in 2015, and we strongly argued that type 2 was better.
Mm-hmm.
And then we wanted to do it in a more precise way. And I think we really overstated the case in the 2015 paper, is why I wanted to publish this one. I wanted to walk it back.
Right. And also in C1, I don't know if shunning inhibition versus hyperpolarizing inhibition is more prevalent.
Well, I guess I would answer that, Alexandra, by it can depend, right? I mean, the whole hippocampus.
Well, right. Now I'm thinking of my project, obviously, which is the intrinsic hippocampus, no extraneural input.
Right. No, but I remember Carmen asked me way back about the reversal potential, the -7, the -90 or whatever that Katie had used, right? And
right.
Carmen, I don't know if you remember asking me this, and you said, well, most people use -75, right? And my answer to you at the time was, that's why Katie used that, just because that's what they experimentally got, kind of thing. Even though and I mean that assumes structural potential, because I guess that's why I kind of have this sort of feeling these days about, so for the models should be really clear about what's going on relative to the experimental data it's based on. So I mean, so you're like that. So, but not
there is some data showing it was shunting and then they dentate. Peter Jonas.
Yeah, they have. Yeah, so there's. Yeah, I mean, because the context matters, right? Like this whole, which is why we really intentionally took the data, like Katie, when she built the models, from the whole hippocampus preparation per se, not from other fast firing stuff, because all these things matter, right?
Right. I guess there's a way, if you look at the data, like the data Katie used, if you look at them in that specific way, you can figure out whether it's hyperpolarizing versus shunting inhibition. I think we just haven't looked at the data in that way.
Right. It's type 1, though, in terms of the FI curve. So
okay.
From Carmen's perspective, yeah. So then it would be type 1. But yeah, so I guess, yeah. So what I wanted to ask you a little bit about, Carmen, because you kind of had to go fast in the end, is that you showed that Laura Colgan data. I was just a little confused about what the context, so I don't know if you mind going back.
Oh, it was a behaving rodent.
Okay, but then it was showing a single, that's why I wasn't sure what.
Let me go back to it.
Yeah, you had that figure there. Yeah, exactly. Yeah, so it's a recording from, so the black is a recording from a...
Their interspike interval counts.
Okay.
The bins are on the bottom, and the counts are on the left. So it's a Gaussian distribution, and it's got a peak at 11 milliseconds, which is on the order of 90 hertz.
Okay, I see. So that's...
And it's got a second peak, which would mean maybe they're skipping some cycles.
Okay, I see.
Yeah, I did have to go over that really quickly, but the point was just, I just want to get out the point is, do we care about oscillators? And I should have brought up the basal ganglia, because in the synaptic, there's a lot of pacemakers and oscillators in the basal ganglia, so surely we care about them there at least.
I honestly don't understand that criticism. Like, do we care about couple? Like, I mean, to me, like, okay, I think those kind of statements by whoever, like whether it's for or against any kind of couple oscillate, like to me that's not a relevant question, quite frankly, because it's the biological system, right? So it's sort of like what we want is theoretical insight to help us understand the biological system. So to argue about, there's no such thing as coupled oscillators or fluctuation, it doesn't make any sense in my opinion, right? Because it only makes sense when you're now talking about, so we're talking about the hippocampus and these recordings and this, we're talking about the basal ganglia, whatever, in this situation, then those questions actually have some meaning. But to just sort of say those statements, that's just, it's just, being closed-minded. Sorry, I'm not online with everybody, so I would say it more strongly than in public kind of stuff. But because we're all trying to figure out these complicated biological systems, right? And the question is, if we're actually trying to do that, let's be clear about what it is we're talking about and how these, analyses or way of looking at the data or like assumptions could be completely clear in the mathematical model. You don't have to like guess what's going on. You know what's going on. You know what you put in. You know what you assume. No issues there, right? It's only like, is it helping us understand? What can we take to predict, to understand that we can go back and test, right? So that's why, your stuff is always good, great in that perspective, right? Because you always, you're dealing with experimentalists, you're looking at it that way and stuff like that. My only thing, my specific thing is about this type 1, type 2 business, right? Because it's hard to, at least nowadays, I feel like biologically, experimentally speaking, it's really hard to kind of be clear about that, right? So that's why this recent work of yours, where you sort of a clear type 1, type 2, you know, I want to read it a little bit more closely. Maybe we'll do that in lab meetings sometime to just really see how that can help us understand, you know, from the biological data, so like the shunting or the hyperpolarizing inhibition that Alexandra was referring to, and things like that, right? Or if there's something in theta gamma coupling, because, you know, Alexandra's going to graduate soon, and I mean, I won't talk about the details of her work right now, but, you know, you'll hear about it as it gets clear. You know, I mean, it is clear to Alexandra, but that's a whole other conversation kind of stuff. But the point is, it's always these, you know, in any of these choices we're making, right? It's not a perfect model or perfect theory, right? It's like, what is it helping us understand, right? So anyways, all I'm trying to say to you is, you don't be offended by people who want to make those kind of statements.
Well, yeah, it was just really, it was a different, one person said that, and then the other person had completely different criticisms. But they wanted me to redo it all for EI, but they were happy with the oscillators.
This was eNeuro, right? Because eNeuro has that consultative peer, you know how Eli has that consultation between the reviewers? eNeuro, I think, does that also, right? So, so did the editor...
And they said they were going to publish the response, but they didn't.
Well, yeah, but did they, you know, if the reviewers had two different kind of criticisms, what you got back from the editor about what you needed to do was...
Well, the editor was Upinder Bala.
Oh yeah, Upi. Yeah.
And in my letter to him, I said, look, there's no way that I can satisfy both of the reviewers.
So what happened in the end?
I wrote like a really long reply, and they accepted it.
Okay. Yeah, because actually, Alexandra, this is remind me of the paper that you had with Katie, right? So 2017 eNeuro paper.
Oh,
so
yeah, in vitro theta is not interesting. That's
yeah.
Oh, that was my paper, I think. Not.
No. Well, okay, I can't remember. It's all a blur now. But I'm talking about the Katie. So it was
I think both of us in both papers we got that criticism. In vitro theta is not interesting. Full stop.
Yeah.
We don't read into the paper.
Yeah, it's not real. It's not real.
Actually, John White, he published with Sylvain Williams on that intact hippocampus.
Yeah, I think it was subiculum, right?
Yes.
Yeah.
And those were the subicular interneurons that in that slow oscillation, and some of them are going at ripple. But now I have no data to know whether they're synchronized with anything. It's just they got one neuron per animal.
Okay. Yeah, no, it's hard.
But I do know that some of them were firing at 140 hertz.
Okay. Yeah. I mean, you see, you know, so there's so many different things to worry about, and I mean, there's so much limitations with experimental data, right? And so that's why I like these sort of, you know.
Are you going to continue to publish on the intact hippocampus?
Well, Alexandra's next couple of papers will be on that. Like, we're working on finalizing one right now, and so once we submit it soon, it'll go on bioarchive, so you'll be able to, like, I mean, Alexandra, I'm assuming we'll put it on bioarchive when we submit it, right?
Yes.
So you'll be able to see it right away. But
it's on theta gamma rhythms, all intrinsically generated, and it's using this model developed in Stanford by Marianne Bezier, like this very detailed model.
Yeah. I've met her.
Right. Yeah, so it's using that model to kind of understand theta rhythms, and they're coupling with gamma a little bit better.
Yeah,
that's my work.
Is that the one that requires the supercomputer?
Yes.
Alexandra's been doing a lot of supercomputer work.
It requires many supercomputers if you have them available. I'm joking, but yes, it's been a thing.
We just got an NSF grant to get a high HPC cluster.
You mean like your own cluster or?
My institution.
Oh, okay. Oh, wow.
I'm co-PI. It's an NSF grant.
Okay, great.
However, the PI quit, and I don't know what happened because...
Who's the PI?
Mohammed Qayyum. He was like a computer science guy, but the point is he quit, and the NSF program officer was mailing him for a month and never got any response. So I don't know how that can happen. It should have bounced or something. So finally he reached out to me because I was a co-PI. And so I reached out to the computer people, and so hopefully we're still going to get it, but they ignored the program officer for a month.
Oh my goodness.
Oh, God.
Well, I mean, I wonder if it's because of, you know, these unusual times, right? You know, I don't know this person.
When somebody quits, they should turn off their email.
I mean, that's what I'm saying. This is very unusual kind of stuff. Yeah, but you know, I mean, what's the time actually?
It was in the middle of our stay at home when he quit, though.
Okay, yeah. But you know, I get what I was going to say in terms of the Sylvain, like in earlier days, like every time they tried to publish something on this intact whole hippocampus, they would always get pushed back. Now this is experimentally.
Yeah, John White says he's not doing it again.
Yeah, so I remember talking to John, this was a couple years now at CNS, and he was saying the same thing, right? And so I guess all I would say, and I think I might have said something like this to him at the time, is that, you know, if you actually want to have some kind of cellular-based understanding, it's very hard once you kind of go too large, because then it's hard to kind of understand. And I've learned so much from Alexander, because Alexander's actually doing like real LFPs kind of stuff, right? Because,
yeah, I mean, for instance, there are ways to bypass that. I mean, you can still use the same model and just use inputs to that model that represents extra CA1 inputs.
Sure.
And
Yeah.
in vivo model, and you get away with that issue kind of thing.
But it's still a lot of computation kind of stuff.
It is, yes, and you don't disentangle what happens intrinsically versus what happens because of external input. So that separation, I think, is important to happen at the first earliest stages. It's important to be clear what happens intrinsically and what occurs because of additional inputs. Otherwise, we will never really know.
Yeah, exactly, and that gets back to the experimental data. So I don't know. I know John was working at Subic White, subiculum type stuff and different things, but it's just, you know, I just mentioned these different cell types fast firing. It's just I don't know what is, you know, how best to fill in the blanks, you know, like with both the experimental and the modeling together. So, you know, you'll see Alexander's paper soon. Like, I guess I'm a bit of a bottleneck right now. Getting the discussion, the next version done, but but basically, you know, if it sort of holds water in the long run, like, you know, maybe it's a way to link these different ideas and stuff, you know, because all I know, and Jeremy knows this, I'm just like, I just really want us to take as much advantage of any kind of theoretical insights and stuff we have out there, because we need it, right? And it's just that not all theoretical people have that mindset. They just kind of will make comments like what you got on your paper, you know, don't want to sort of worry about the biology or just wave their hands and say whatever kind of stuff. I don't have time. Personally, I don't have time for those people, right? So because it's not helpful, right? If you don't want to bother to even care about the biology in some capacity, right? Or at least, you know, if you don't, and then it's just, you're just in a, you know.
It's actually, I have 3 physicists in my lab, and Ruben is no longer in my lab, but he was also a physicist.
What is he doing?
He's at George Washington University in DC. He's got a research scientist position.
Okay.
But anyway, they're all physicists.
Okay.
And it's just, in many cases, it's just a real struggle to get them to understand what the equations map onto in the biology, and that the biology is messy.
Yeah, I think some people just, you know, don't want to worry about the messiness, or I don't know. I mean, I guess I see it as a very individual thing. So Jeremy, you know Jeremy, right? Jeremy Lefebvre, who's
the name is extremely familiar.
Yeah, so he's an affiliate scientist here at the Crumble, but now his main position is in Ottawa. So he has that, but he's a physicist also. But I mean, he's interested in sort of the stimulation and like sort of the different level, right? So he's Poisson type equations, but we have fantastic conversations, and it's very helpful because I don't deal with those kind of models, but I want to understand why people do certain things. That's the stuff you don't read in the papers. Like, what is it? Why would you do it this way? And what's the advantage and blah, blah, blah kind of stuff, and what's the limitations? And similarly, of course, he cares about the cellular stuff. He's just personally not doing those kind of models, right? So it's like, how can we meet, right? But he cares, or he's interested in his models doing or showing something, right? You know, like whether it's in terms of, you know, epilepsy or whatever, right? Like, so he by default cares about the biology, but he's a theoretical physicist type person, right? So I think it's a very individual person type stuff. Like, so so I mean, I've had a few people in the past, math type people, who just could not deal with the messiness of biology. Let's put it that way.
I had one. I actually had let a postdoc go right at the same time that Ruben quit, and so I was really shorthanded for a while. But and my postdoc that replaced Ruben, Gillum, he's in Barcelona.
So
I can't get him here.
Oh, right now you mean because of
he had a job offer, but he can't get a visa.
Right. So you're just working virtually, or he can't start?
Yeah, but I can't pay him. I need to talk to my provost again.
Okay.
It's just that's a very difficult situation for a state institution to do a subcontract to an individual in another country that doesn't have a social security number, and they can't drug test.
Oh my gosh. Oh gosh, complications.
Anyway, that's just a logistical hassle.
Yeah. No. I mean,
coronavirus. I mean, I had a really hard time filling my open positions. And, you know, not to be able to get him physically here is a bummer.
Yeah, yeah. Well, I think I might have reached out to you that we had this job, you know, another job, a computational neuroscience job here at Krembo, right? And so we have our short list now, but then because of coronavirus, travel shut down, so we had to put it on hold.
Right.
So yeah, but anyways, hopefully we'll get, well, we're just now starting to open up. So anyways, that's the whole part. So yeah. Alexandra, did you have other questions?
No, I'm good. Thank you very much.
Nice to meet you.
Dr. Canavier, very nice to talk to you again. I was there in your talk last year at CNS, and I enjoyed it very much. And now it's a bit of a revision for me.
Was it in Barcelona?
It was in Barcelona, yes. At the workshop, the oscillations workshop.
Right. That was Horacio.
Horacio, yes.
He gets very upset if I say it wrong too.
Anyway, it's an easy name. Horacio is an easy name. Like,
but I think in English it might be Horacio or something. I don't know. However I said it, he didn't like it. But
I mean, he's like
he's an old friend. He's an old friend.
Great. Yeah, he's like than I am.
Curly hair. He has no hair. He's bald.
That's true. That was a long time ago. That curly hair thing. That was a while back.
Yeah, but actually, who initiated? Was it you or Victoria or Avrama? It was one of you three who started that. I can't remember who it was. Would have been Avrama?
Leslie was there. Leslie from Chicago.
Yeah. Leslie Key.
Yeah, she was there. She
yeah.
curly hair.
No, he was very mad because no one believed that he was Spanish.
Oh.
Because he's very white, right? Whereas with me, everybody. Sorry?
I thought he was from Brazil. No?
Right, that he speaks Spanish. Yes, probably he's not from Spain, but he speaks the language. But nobody would speak Spanish to him. On the contrary, everybody would speak Spanish to me, and nobody would believe that I don't speak Spanish, no matter how many times I said it to them.
I think it's Chile he's from, right?
My father's from Chile.
Oh,
well, he was.
Yeah, I'm pretty sure. Well, you should tell that to Horacio. I would have to double check, but I'm pretty sure it's Chile.
And in fact, I've been to Chile several times recently. I have a collaborator in Santiago.
Oh, okay. There you go. Because there's some institute there. Because he has some position there, some math. Okay, now I'm no. Oh, sorry. I think it's Argentina. Okay.
The Argentina rings a bell.
Yeah. Okay, sorry.
I was wrong about Brazil.
Yeah. It's definitely not Brazil. I know that.
Because that's Portuguese anyway.
Yes, that's right. No, we have a good friend in Brazil.
Nice. Okay, thank you so much.
Well, thank you all for asking. It was really nice talking to you, and I am definitely going to go look up that paper right now. It would have been very handy to have in a rebuttal to the reviewer.
Yes, and also, your talk helped me understand very much, again, why those pyramidal cells don't fire at every theta cycle. This is something that I've seen in theoretical studies, but this PRC analysis makes it more clear to understand why it's actually happening. But yet it seems like with the pyramidal cells, for example, well,